Welcome medicos to yet another video on this channel. Curious about how your brain controls every movement? In this video we'll unlock the secrets of spinal motor systems, explore key neural pathways, and understand how brainstem control impacts your posture and reflexes. But what happens when these systems fail? From sudden loss of movement to life-threatening conditions like decerebrate rigidity, understanding these mechanisms is vital for preventing and managing severe motor disorders. Imagine a bustling city, with information flowing from countless sources, converging at a central hub. This is the essence of convergence in the spinal cord, where multiple neural inputs converge on a single motor neuron. These motor neurons, the final common pathway, receive a symphony of signals from various parts of the nervous system. Among these inputs, the signals from muscle spindle group IA afferents are especially important. These sensory fibers, nestled within our muscles, act as biological strain gauges, providing constant feedback on muscle length and stretch. When a muscle is stretched, these IA afferents fire, sending a surge of excitatory signals to the spinal cord. These signals converge on alpha motor neurons, the nerve cells responsible for commanding muscle contraction. The greater the stretch, the stronger the barrage of IA afferent activity, and the more likely the alpha motor neuron is to fire. This mechanism, known as the stretch reflex, is essential for maintaining muscle tone, posture, and our ability to react quickly to changes in muscle length. Understanding convergence is crucial for medical professionals. It helps us decipher the complex interplay of signals that govern movement and provides insights into potential therapeutic targets for restoring motor function. Imagine a group of friends trying to push a stalled car. Individually, their efforts might be insufficient, but together, their combined force can get the car moving. This is the principle behind spatial summation, where multiple inputs, arriving at different locations on a neuron, combine to produce a stronger effect. In the spinal cord, spatial summation plays a crucial role in muscle activation. Each muscle is controlled by a pool of motor neurons. A single motor neuron may receive inputs from hundreds or even thousands of synapses, each carrying a tiny electrical message. Some of these inputs are excitatory, pushing the motor neuron closer to its firing threshold. Others are inhibitory, pulling it away from that threshold. Spatial summation refers to the way these excitatory and inhibitory inputs are added together over the surface of the neuron. When the combined excitatory inputs outweigh the inhibitory ones and the overall membrane potential reaches a critical threshold, the motor neuron fires, sending a command to the muscle fibers it innervates, resulting in muscle contraction. This delicate balance of excitation and inhibition combined through spatial summation allows for fine control over muscle force. Imagine a drummer playing a snare drum. A single tap produces a brief sound. But if the drummer strikes the drum repeatedly in quick succession, the individual sounds blend together, creating a sustained, louder sound. This is the essence of temporal summation, where the timing of neural inputs plays a crucial role in determining the output of a neuron. In the context of the spinal cord, temporal summation refers to the way that rapid, successive inputs from a single synapse can summate, increasing the likelihood of triggering an action potential in the motor neuron. If a single excitatory input from a muscle spindle IA afferent is not enough to reach the firing threshold of a motor neuron, a rapid succession of inputs from that same afferent can summate over time. This temporal summation can push the membrane potential past the threshold, triggering an action potential and leading to muscle contraction. The closer together these inputs arrive, the stronger the summation, and the more likely they are to elicit a response. Understanding temporal summation is crucial for comprehending how the nervous system encodes information not only in the strength of signals but also in their timing patterns.